Welcome to the first video on cell cycle. Self reproduction is characteristic feature of all cells, and it can be also said for all living organisms. A single bacterium to form a colony consisting of millions of progeny cells, it takes only an overnight. Repeated cycles of cell growth and division results in development of a single fertilized egg into approximately 10 to the power 14 cells that make up human body. So, what is a cell cycle? A cell cycle is a series of events that takes place in a cell as it grows and divides. So, the division cycle of most cells consists of four coordinated processes. One is cell growth, that is the continuous process. Next is DNA replication, that is represented by the S phase. Next is distribution of the duplicated chromosomes to the daughter cells that takes place during the M phase. And finally, division of the cell into two daughter cells in the that is known as cytokinesis. So, what are the different phases of cell cycle? If we take an example of human cell cycle dividing in culture, we can see a, it totally takes 24 hours. So, normally, cell cycle can be divided into two basic parts: mitosis, that is nuclear division, which in human cells takes 1 hour, and interphase, that takes 23 hours. So, M phase is represented by mitosis, which is usually followed by cytokinesis. In between the M phase and the DNA replication phase, there is gap 1 phase. So during gap 1 phase, the cell is metabolically active and continuously growing. In human, it takes about 11 hours. And then it is followed by S phase, that is synthesis phase, in which DNA replication takes place. In human, it takes about 8 hours. And it is next followed by the gap 2 phase, where the cell continuously grows and proteins are synthesized in preparation for the next mitosis. So, we can see that all the cells do not follow the same rule. For example, in embryonic cell cycle, the time is very less. The cell cycle takes about 30 minutes, and it is devoid of any gap 1 and gap 2 phases. And you can see that cell is not growing, it is only dividing to form the embryo. In contrast, the adult animal cell ceases division altogether, for example, in nerve cells and fibroblast cells. And many cells, they only divide occasionally as needed to replace that have been lost due to injury or cell death. So it enters into the G0 stage. How do we identify the cell stages? Identification of S phase cell is can be done by incorporation of radioactive thymidine, as you can see in this picture, or it can be done by biochemical test, for example, a flow cytometer, which relies on the fact that DNA content varies during the cell cycle. So, throughout the G1 phase, the DNA content is 2N. After it starts DNA replication, the DNA contents increases till it reaches G2, it reaches 4N. So in G2 and M phase, the DNA content remains 4N. So by seeing whether the amount of DNA present per cell is 2N or 4N or in between, we can determine the cell phases. So, with this, we have learnt about what is cell cycle, what are the different phases of cell cycle, and how DNA content varies with cell cycle phases. In the next video, we will learn about cell cycle regulation.